is a conserved N-terminal protein structure named after CRAL-BP and trio guanine exchange factor. So if you're familiar with our lab, we study the evolution of vision. And so why is this gene family relevant to us? Well, it's named after CRAL-BP, which stands for cellular retinaldehyde binding protein. And so here's the CRAL-TRIO domain that I talked about. And proteins that have this domain transport lipophilic molecules. In this case, this protein transports 11-cis-retinol. So it transports 11-cis-retinol to photoreceptor cells where it binds an opsin protein. And when you have light, 11-cis-retinol changes its configuration to all-trans-retinol, and then you have phototransduction cascade, which is the way in which light is converted into electrical signal, and this is how we detect light. In humans, mutations in a gene encoding CRAL-BP result in inherited blindness known as retinitis pigmentosa. This is degeneration of the rod cells. And in insects, these CRAL-TRIO domain proteins exist, but their function is not really well known except for in Drosophila. A gene known as PINTA encodes a protein that binds 3-hydroxyl 11-cis-retinol. And so this previous study, where they looked to see where PINTA mRNA is expressed, and they found it in the retina, but also in the optic lobe and in the brain. So the Drosophila compound I is made up of 800 ommatidia that have a structure that looks something like this. You have 8 photoreceptor cells, R1 through 6, and then R7 and 8. And R7 sits on top of R8. R7 expresses either of 2 UV opsins, and R8 expresses either a green or a blue wavelength opsin. R1 through 6 express RH1. So when you have a mutation in the PINTA gene, this results in a decrease of RH1. And so then this PINTA gene is involved with chromophore transport in Drosophila. And a previous study in our lab sought to create the first insect phylogeny of this gene tree. So they looked at reference genomes that are publicly available, and they used Manduca, Danaus, Bombyx, Tribolium, Apis, Anopheles, and Drosophila to search for these genes and then create a phylogeny of them. And so what they found was that there was a lot of conserved amino acid residues, and what they also found was there was lineage-specific duplications of these genes. But what was really interesting is there's an expansion in Lepidoptera, so this is moths and butterflies, and this expansion results in twice as many CRAL-TRIO domain genes in these moths and butterflies in comparison to these other insects. So the questions that I try to address in my study is, how are CRAL-TRIO genes evolving in Heliconius melpomene? By adding another butterfly, we can confirm or reject that this lepidopteran expansion is actually occurring. And we also use Heliconius melpomene because we have a reference genome, and then thanks to Christigan's lab, we have a lot of resequenced genome where we can see how these genes are present or absent in other individuals. The next thing I wanted to look at is, what are the expression patterns of these genes in different sexes and tissues? So when we did the previous study, we did not find a homologue for PINTA. And if we wanted to see where these genes, which of these genes is a candidate for chromophore transport, then presumably it would be upregulated in the heads. And the Manduca project did have RNA-seq data for the heads, but only for the heads, so we cannot compare it to other tissue and see if it was indeed upregulated. So the objective of this study is to identify a CRAL-TRIO gene potentially transporting the visual pigment chromophore in butterflies by using Heliconius melpomene. And I made RNA-seq libraries, and I made a de novo assembly in Trinity. And then I used RSEM to map each of the libraries back to the de novo assembly. I used EdgeR to do a whole transcriptome-wide differential expression analysis. And then I used FPKM to look specifically at these CRAL-TRIO genes to see how they are expressed in the different sexes and tissues. And once we've identified a candidate gene that's upregulated in heads, I used RT-PCR to see whether it's expressed in eye or brain and at what levels. So for my results, here is a branch of the tree. I'm not going to show you the full tree because it's a little bit overwhelming. But as you can see, this is Tribolium, this is a beetle, and this is the Lepidopteran expansion. It holds true for Heliconius. Heliconius is here in red. We do see that there's a lot more of these CRAL-TRIO domain genes in Lepidopterans compared to other insects. We found a total of 44 CRAL-TRIO domain genes in the reference Heliconius melpomene genome. And this is comparable to Manduca sexta, where they found 42 CRAL-TRIO domain genes. And also, I named each of these Heliconius genes in the order as to which they appear on the scaffold. And if you look closely to genes that are closely related on the tree, they're adjacent to each other. And so we found a lot of tandem duplications, suggesting that there's an array of tandem duplications for these trees in the Heliconius melpomene genome. And so this is important because when you have a gene duplication, what you expect to happen sometimes is one of the duplicates becomes pseudogenized and acquires mutations. But you can have retention of the two gene copies, even if you have two of them, if you have neofunctionalization. This is one of the gene copies acquires a new function. You can also have the two genes retained if you have subfunctionalization, where each of them shares in the ancestral gene function. But before you have neofunctionalization and subfunctionalization, you have copy number variation. And copy number variation is important because from studies by Macias-Mu, animal chemosensory receptors have a large variation in the number of chemosensory genes between species and within species. And they suggest that this might be happening due to drift, but that these copy number variations are adaptive in a new niche. And in addition, our lab, using Heliconius melpomene, looked at gustatory and olfactory receptors, and they found that these receptors had undergone a lot of duplications, and they have copy number variation, suggested they're undergoing rapid evolution. So we wanted to see if the CRAL-TRIO domain genes are like these chemosensory genes undergoing copy number variation, since we have a lot of duplications. So here are the 44 CRAL-TRIO domain genes on the scaffolds of the new Heliconius genome. And alternating black and white boxes are the different scaffolds. So here is an example of genes that are tandemly duplicated, and a really large expansion occurs here in 15 through 28. All of these genes are very close together, just in tandem duplications. And so then I took the 18 resequenced genomes and mapped them to the reference. This was from 4 different subspecies. So 4 Heliconius melpomene, 4 rosina, 4 amaryllis, and 4 gallopy. And I looked to see if they had copy number variation between these genomes and the reference assembly. And I found that, so if you look at the genes mapped on the chromosomes, the ones that are not shaded in do not have copy number variation, and the ones that are shaded in have copy number variation. So we found that 26 of these CRAL-TRIO domain genes have copy number variation. And next, I was looking at RNA-seq. So I took RNA from the antennae, the mouth parts of Heliconius melpomene, males and females, and mouth parts include proboscis and labial palps. And then whole head, and from the 6 legs. involved in vision and phototransduction. And then I looked for the CRAL-TRIO domain genes to see if they are found within these upregulated genes, and I found only one,